Hi, my name is Kimberly Rodriguez, and this is my photosynthesis project. So photosynthesis comes from plants. So in a plant, we have plant cells, and within these plant cells, there's our chloroplast. And within these chloroplasts, there are little stacks of alkaloids, and each stack of alkaloids is actually a grana. But each individual alkaloid is filled with lumen, which is going to be a alkaloid lumen. That's going to be inside the alkaloid. And then outside the alkaloid, which is inside the chloroplast, we have stroma. And so we move on. So these alkaloids each have a phospholipid bilayer. And we've learned that these phospholipid bilayers are able to hold proteins. And here we see there are 4 different protein complexes that this alkaloid membrane is holding. So how photosynthesis starts, the sun sends down the UV rays, the light rays, and within these light rays there are photons. These photons are actually absorbed by chlorophyll, and these chlorophylls would be inside the first protein complex, which is photosystem 2. So photosystem 2, what it does is it absorbs the electrons from the photon and it energizes it up. It gets it all riled up, and the electrons then makes its way towards a reaction center chlorophyll. And then once it's all riled up and ready to go, it'll push the electron to an electron transport chain. And through the electron transport chain, different proteins will pick it up and take it throughout the cycle, or take it throughout the chain. And these electrons, and during the electron transport chains, electrons loses the energy in order for everything else around it to capture the energy to keep everything else going. So this electron gets all riled up in photosystem 2 and is pushed out and is carried by this PQ molecule, which is actually plastoquinone. And this plastoquinone accepts the electron, accepts the excited electron from photosystem 1 and makes its way towards the second proton complex. You see here, which is actually cytochrome B6F complex. Now, as that electron is moving on, the chlorophyll inside the photosystem 1 realizes that the electron is gone and feels that it needs to be replaced. So what it does, it actually oxidizes a water molecule. It oxidizes oxygen, basically, and it takes the energy, the electrons from the water molecule back into photosystems 2, and it splits the water, separating it into protons and oxygen, and that's how oxygen is made. And now these protons are inside the lumen, inside the alkaloid. So as this electron makes its way to the cytochrome B6F complex, this complex actually uses the energy from the electron and brings in protons from in the stroma in through the alkaloid membrane into the lumen, and that actually creates a concentration gradient. So now we have all these protons within the lumen. There's going to be more than one. It's actually very exaggerated. This is happening all around. So now that there's so many hydrogens inside the lumen, another protein complex, which is ATP synthase, realizes that there's a high concentration of protons inside the alkaloid, that it starts working and pushing, and it starts to push those hydrogen atoms out. And as it pushes it out, it actually turns and phosphorylates ADP into ATP, and that pushes it out back into the stroma. So now we have all these hydrogens coming in, and the ATP synthase pushes it back out and phosphorylates ADP into ATP, and that's outside into the stroma. So as this electron reaches, once it's done here at the cytochrome B6F complex, it's all out of energy. It's already wasted all the it's gone down its energy state. So it gets sent through another electron transport chain, and this time it's picked up by here, which is this one, which is PC, which is actually photocyanin, plastocyanin. So this plastocyanin electron carrier accepts the electron from the cytochrome B6F complex and reduces it and gives it to photosystem 1. Now when this electron reaches photosystem 1 is actually already also getting hit by sunlight, so it is receiving more protons, and what it does is it takes this electron and revives it, re-energizes it, gets it all riled up again to send it through another electron transport chain. This time it's going to go up towards out of the alkaloid membrane. So this electron gets sent up to one last electron transport chain, and it gets picked up by this DFD protein right here, which is actually ferredoxin. And ferredoxin is the last electron carrier, and it accepts the electron from photosystem 1, and in order to keep it going, it is catalyzed by FNR right here. This enzyme comes in and catalyzes the electron transport chain, and this FNR is ferredoxin NADP reductase. So FNR catalyzes the oxidation of the ferredoxin molecule, and what it does, so now that electron is being used up to make NADP plus into NADPH. So now we have the electron that was accepted from these protons, it's made its way through, and it's sent through another electron transport chain. So this ferredoxin is catalyzed by FNR, and it turns NADP plus into NADPH. So now this light dependent reaction, this is the light, and this all has to happen with light. It depends on the light to happen. So now this light dependent reaction has made NADPH and ATP, and it's pushed that out into the stroma. And once that hits the stroma, those actually go into the light independent reactions, which is called the Calvin cycle. Now here the Calvin cycle is light independent. It does not need light to do it, but that doesn't mean it happens in the dark. It can happen at any time. So the Calvin cycle happens in 3 different steps. The first step is actually the beginning and the ending step, because it's a cycle, it just keeps going. It is where carbon dioxide is actually fixated to RUBP, which is ribonucleus biphosphate. And this happens. CO2 is fixated onto ribonucleus biphosphate by ribisco. Ribisco is an enzyme. Well, the full name is RUBP carboxylate oxygenate, and that sticks the CO2 onto the RUBP. So what happens is once it's joined together, it becomes unstable and it freaks out to where it breaks in half. So it cuts it in half, and now you have a 3-carbon molecule, which is going to be 3-phosphorylate phosphoroglycate. Right. And so these phosphoroglycates actually goes on to step 2, which is reduction, and it is reduced by ATP molecules and the NADP molecules that are released from the light-dependent reactions. And it goes down to be reduced to 3GP. Now this 3GP is glyceride 3-phosphate. Now these glyceride 3-phosphate are actually, well, you need a few of them. And actually the final step of the cycle, these these 3DPs are actually reused to go back up and to make RUBP again. But in order for all this to happen, a certain number of cycles need to happen. So what you need is, you're going to need 3 carbon dioxide molecules to be bounded to 3 RUBP molecules to make 6 3-phosphorylate molecules. And then you use about 6 ATP molecules and 6 NADPH molecules to make 6 G3P molecules. Now you only need 5 to 5 of these 3GP molecules go back up into the cycle to make 3 RUBP molecules. The other one, since there's one left over out of the 6, this one actually goes out and it makes glucose, and it can make fructose or sucrose or anything else that it needs for the cell. Overall, you need about 9 ATP molecules and about 6 NADP molecules, and that's how we make sugar and energy for the cell. Thank you.